I wanted to just start out by thanking the network for supporting this research. It's been wonderful to have some of the mentorship that's been part of the network, as well as, of course, the financial support. And then I also wanted to just take a moment to acknowledge the collaborators on the work that I'm going to be presenting today. So in particular, Steve and Steve's team, who did all of the genomics work and the bioinformatics that I'm going to be presenting today. And then also two of my collaborators who aren't here, Payman Hamadi, who's very interested in the role of macrophages in myeloma, and his lab did all of the collection of the macrophages and the monocytes that we collected for this study. And Natalie Callender, who's a clinical hematologist who specializes in myeloma, and she did all of the work with figuring out the clinical correlates that we used, and they've been very supportive of this kind of work as well. All right, so for those of you who aren't familiar, multiple myeloma is a cancer of the plasma cells. It's a recurrent cancer that is treatable but rarely curable. The median survival time for myeloma after a diagnosis is about 4 to 5 years. Autologous stem cell transplantation can extend survival for myeloma patients, typically by about 2 years. It allows patients to receive a very high-dose chemotherapy that would be lethal because it ablates the bone marrow. But the patient is then rescued by the infusion of his or her own previously harvested stem cells. It's a rigorous therapy with significant morbidity for patients. And while little is known about biobehavioral influences in stem cell transplantation and also in myeloma, there's emerging research from our lab, as well as Jennifer Knight's work and a few others, suggesting that stress-related psychosocial factors may influence the immune recovery after transplant, particularly in autologous stem cell transplant. And this is important clinically because a more optimal immune recovery after transplant not only prevents post-transplant infections, which are a significant source of morbidity after a transplant, but there's also evidence that a more optimal immune recovery is associated with reduced risk of relapse and a longer survival. So in this study, we looked at gene expression patterns that might underlie the relationships that we're observing in other work between stress-related psychosocial factors and immune recovery after transplant. We've focused on gene expression of the monocyte precursors of macrophages because these cell subsets influence the recovery of adaptive immunity following HSCT. So in particular, monocytes and macrophages can serve as a bridge between the recovery of innate and adaptive immunity. And alternatively, activated M2 macrophages, which are distinguished from the classical M1 macrophages, can suppress the adaptive immune response following a transplant. And also, as many in the network research that has been going on, showing that macrophages can promote tumor development. And that's also true in myeloma. So there's been quite a bit of work showing that macrophages promote angiogenesis and protect myeloma cells from apoptosis, as is the case with other types of cancer. And work by my collaborator, Payman Hamadi, has found that macrophages can stimulate the proliferation of myeloma cells and also increase secretion of cytokines that are important in myeloma pathogenesis. So the objective of our project was to examine the extent to which stress-related psychosocial factors predicted gene expression patterns of monocyte precursors of macrophages among multiple myeloma patients who were recovering from an autologous stem cell transplant. The sample included 26 adults who were receiving autologous transplants for multiple myeloma at our cancer center. myeloma is somewhat more common in men and also increases with age, so our sample reflected those characteristics. The median age for myeloma is actually quite a bit older, more like 70 years old, but we tend to transplant younger individuals. So this data was, or the project was done in the context of a larger study that our lab was doing, where we did a variety of psychosocial measures prior to transplant, including measures of depression and social support. And for this work, we were particularly interested in the social attachment subscale of the social provision scale, which looks at the extent to which people perceive that they have close personal relationships. And then also prior to transplant, and again 1 and 3 months after transplant, we collected blood samples from the participants. And Dr. Hamadi's lab isolated CD14 positive cells from peripheral blood using magnetic bead separation. And then we then sent our samples to Steve for genome-wide transcriptional analysis and promoter-based bioinformatics to determine the gene expression patterns. The primary analyses focused on a 53-gene conserved transcriptional response to adversity, or CTRA profile, that many people here are familiar with. It's a profile that's commonly seen in adverse life circumstances and stress in a variety of contexts and populations. And it involves upregulation of pro-inflammatory genes, which in our context, myeloma, are important because of the role of inflammation in myeloma pathogenesis. And then downregulation of type 1 interferon, antiviral, and antibody-related genes. And so, again, important in our context with respect to immune recovery and preventing infection following a stem cell transplant. So the first set of analyses used mixed effects linear regression models to examine the extent to which depression and social attachment predicted CTRA expression across the assessment points, 1 and 3 months post-transplant. We didn't see any significant relationships between depression and CTRA expression. But we did find that patients who reported greater attachment showed downregulated CTRA expression 1 and 3 months post-transplant. The minimally adjusted model included covariates that were associated with CTRA, which were time since transplant, sex, and presence of high-risk cytogenetics. And the fully adjusted model additionally covaried for age, disease stage, number of prior treatment regimens, and also whether the patients were on maintenance therapy. And again, Natalie Callender was really instrumental in sort of identifying what the important clinical factors that we needed to consider were. So what this figure shows is that participants who scored high on attachment showed a 38% reduction in CTRA expression in the minimally adjusted model and a 33% reduction when we fully adjust the model relative to low-scoring patients. The follow-up analyses then examined cellular and molecular mechanisms for the differences we were seeing in CTRA gene expression for the participants who had high versus low support. And the first step was to identify genes that had at least a 1.25-fold difference between those who scored high and low on support. And next, transcription element listening system promoter-based bioinformatics analyses were applied to these genes by Steve, and these analyses test whether the pattern of genes identified indicates a difference in transcription control pathways that might mediate gene expression. So kind of going down the list here, what we found, we didn't find any significant relationship between social attachment and the glucocorticoid receptor at the top there. Social attachment was associated with an unexpected increase in expression of NF-kappa B, which, as many here know, is pro-inflammatory and can be tumor-promoting. But we did see a more expected increase in expression of interferon response factors, which, again, are important in antiviral responses and can also inhibit tumor growth. And then we also saw decreased expression of cAMP response element binding protein, which marks beta-adrenergic signaling. And then the final set of analyses examined whether there was a link between social attachment and shifts in the monocyte population. And here, transcriptome representation analyses were applied to that same group of identified genes that showed a difference between those who scored high and low on social attachment. And these analyses tested whether the pattern of differentially expressed genes indicated upregulation of specific monocyte subsets. And what we saw there with the TRA was that patients who reported higher attachment had increased prevalence of the more mature tissue-healing CD16 positive monocyte transcriptome. We didn't see a relationship with the less mature CD16 negative transcriptome. So to summarize the findings, we found that multiple myeloma patients who reported greater social attachment prior to transplant, post-transplant then showed upregulation of genes and associated cellular and molecular pathways that foster adaptive immunity. And again, that's critical for preventing infections and optimizing immune recovery following a transplant. And these genes and pathways are also associated with inhibition of tumor growth, and that is very critical in the myeloma context given the recurrent nature of this disease. Attachment was also associated with downregulated expression of genes that promote inflammation, and again important because of the significant role of inflammation in the growth and development of myeloma. But the links with the cellular and molecular mechanisms associated with inflammation were not as consistent and robust. So to conclude, our findings highlight pathways by which behavioral factors might affect gene and cellular mechanisms that can influence treatment outcomes for multiple myeloma patients. And again, I just wanted to acknowledge funding sources, not only the network, but also other NCI mechanisms which have been really important in this work. And I'm happy to take any clarifying questions.